On the reset the button again.
Wait.
Yes, we have the password.
Yeah, first I'd like to apologize. I know that you like to go out and drink and relax. Sorry. I'll try to keep it really light. And in particular, this is really a very introductory lecture, so there will be lots of pictures. It's only a presentation with slides and no equations. So the equations, I'll start writing down equations in the second lecture tomorrow. So the goal today is to provide this introduction on brains, what kind of networks we find in brains, what are the components of networks, neurons, synapses, and also tell you a little bit about learning and memory, how we think learning and memory work in brains. And so more generally, the outline of the course, so there will be 5 lectures. So after the introductory lecture today, I will tell you about a very classical model. It's basically the grandfather of all memory models in neuroscience, is the Hopi model, and tell you about various generalizations of this model. In the next lecture, I'll try to connect the Hopi model with neurophysiology, so for results from neurophysiological experiments and how you can actually implement learning rules that are inferred from data in this framework and what kind of results you get when you do that. In the 4th lecture, I'll talk about the dynamics of networks and how we can understand free recall and also how we can understand the sequences, how we can store sequences in networks. And the last lecture will be about how to optimize information storage in network, and I will talk in particular about a particular approach called the Gardner approach that was introduced by Elizabeth Gardner a long time ago. All right, and so for today, I will go through basically this list. This will be very a very high-level overview. I won't go into any detail of all these things for a long time. So let's start out by a human brain, and so here, and first let's see what are brains made of. And we see that there are basically a number of major structures that compose brains, and the biggest one by far is the cerebral cortex. And the cerebral cortex is involved with lots of our of what we do. It's involved with perception. You have lots of sensory areas. This is the visual cortex here. You have the auditory cortex, gustatory cortex, somatosensory cortex, and olfactory cortex here. But also, it's also involved in our high-level cognitive abilities like decision making, problem solving. And that's the frontal part of the cortex. And eventually, it's also involved in generating movements. This is the motor cortex. Okay? So everything, all these things that cerebral cortex does. But of course, it's not the only structure. There are also 2 other structures, important structures that are shown here, which are underneath cortex. Cerebellum is a very important structure for motor control, motor learning, and also balance, maintaining balance, posture, coordination. And here the brainstem. Brainstem is fundamental for controlling involuntary movement, and very important things like, for example, breathing. The control of breathing is done by the brainstem. But of course here there are many more parts of the brain that we don't see from this picture. So let's look at the brain from other views. So if we look from under the brain, so this is an example of a sensory system, which is the visual system. So as you know, visual informations come through the retina, which is itself a highly complex neural network that's composed of approximately a little bit less than 100 cell types that analyze the visual signal and then send the output to the optic nerve, and it goes to a relay station called the lateral geniculate nucleus in the thalamus, and then it projects onto cortex. So primary visual cortex is the the first area, a cortical area that receives visual information, and there are similarly sensory systems for all the other senses that I mentioned, so the auditory system, somatosensory system, etc., etc. Also, another parts of the brain that we haven't seen so far are deep inside the brain. So now this is inside a view from inside the brain, and we have a number of structures that I will not be detailing all, but let me just mention the hippocampus, which is a very important structure for memory. So we'll talk about it in more detail later. Amygdala is an important structure for emotions and also the emotional content of memories. And there are other structures that I won't be telling you about, like basal ganglia are important for selecting actions, and hypothalamus is important for very basic functions like controlling body temperatures, releasing hormones, and things like that. So, in fact, there are people who are interested in modeling all these different structures. So computational neuroscience has become a huge field. There are many different questions that people try to understand, from sensory processing to motor control. In this series of lectures, I will just be focusing on memory. Okay? There are essentially four major structures that are involved in memory in the brain that are now shown here in this slide. And let me just start with the hippocampus. So, right, I can just read what's on the slide. So the hippocampus integrates information from a number of brain areas to form a single declarative memory that will then transfer to long-term memory. So hippocampus is very important to form the first formation of memories, and it stores these memories in a kind of short-term buffer. Then these memories are transferred progressively in cerebral cortex. And these could be episodic memories, these can be semantic memories, so all kinds of memories that are stored in the cerebral cortex. The amygdala is important for the emotional content of memories, as I've already said, but I won't be really telling you anything about that. And finally, the cerebellum is important for procedural memory. So procedural memory is things like motor skills, but they're also, in fact, now we know that it's more generally involved in storing also cognitive skills. So skills in general, not only motor skills. And in particular, it's crucial for motor skills that require motor coordination, like when you're learning to play piano, for instance, or riding a bike, so you need to coordinate different things on cerebellum. I will come back briefly on cerebellum in the last lecture, but I will be focusing more on what happens in particular mostly in cerebral cortex. Okay. And so far I've shown you only pictures of human brains, but of course most of what we know about how brains work really comes from studies in animals, not humans. And the reason, of course, is that you can do lots of experiments you can do in animals that you can't do in humans. You can't stick electrodes into human brains, except rare exceptions, like epileptic patients, for instance. And so neuroscientists have studied a whole zoo of different animals, and of course, these go from the simplest to the most complex. Here we have the one on the top is C. elegans. So C. elegans is a little worm that's approximately 1 millimeter long, and the brain of C. elegans has just 300 neurons. So that's a really simple nervous system. And this is also the only animal that we actually have the full wiring diagram. We know for each neuron which other neurons it's connected to. And it's the only animal that for which we currently have this whole wiring diagram. And so that's nice because it's very simple, so you can then hope to understand more easily how it works. Unfortunately, of course, it's very far from us, right? So if ultimately the goal is to understand how the human brain works, C. elegans is perhaps a little bit too far. Then you can go down this list and you have more and more complex brains. So Drosophila is the next highly popular animal, and in fact, are because of genetic tools, so you can genetically modify very easily Drosophila. Also, people are making huge progress in reconstructing the brain of Drosophila. So now we actually have the wiring diagram of slightly more than half of this brain. And note that the number of neurons now has gone up by several of the magnitude. You have 100,000 neurons in the brain of this little fly. And then you have other animals. So larval zebrafish has become a very popular system because it's transparent, so you can actually visualize the activity of the whole nervous system, so you can actually see how neurons are active while the animal is actually producing behaviors. So that's very nice. Zebrafinch are very popular because they are because they can learn songs and produce songs. So that's a nice model to try to understand language, right? So because they can learn complex songs with different syllables and so on and so forth. And then we go to mammals, and of course we are interested in mammals for obvious reasons. Mouse is by far the most studied animal, the most studied mammal, and in particular because of the availability of genetic tools, again, you can produce genetically modified mice very easily. Finally, monkeys, and the reason people use monkeys now is because they are the closest to humans, in particular for studying higher level cognitive functions. That monkeys are close to the brains of monkeys are thought to operate in a way that's very similar to us. And in fact, if we just restrict ourselves to mammals, the structure of the brain of different mammals is actually quite similar. Of course, the size and the details are quite different. We see that the rat, we also see a big this is a cerebral cortex of a rat. Of course, it looks quite different than humans, but you will see also roughly the same areas, the visual system here, the frontal cortex on the other side, and so on and so forth. And this shows this comparison with a little bit of detail, where you see that all the major structures that we have, rats also have, but the proportions are very different. Particularly, notice that cerebral cortex is the fraction of the brain that's the cerebral cortex is much smaller in rats than humans. On the other hand, olfactory bulb is huge in the rat, but very small in humans. So in the following, I will show you in particular data from rats, from rodents, rats and mice, and also monkeys. All right. So this was just about the macroscopic structure of brains. Right. So now let's kind of zoom in and see what these structures are composed of. And I will take the example of cerebral cortex. And if we zoom in pretty much anywhere in the cerebral cortex, at some point we start to see that it's composed of neurons. This is approximately a cubic millimeter of cortex. The cortex is basically the cortex of humans. If you flatten it, will be approximately the size of a pizza, and also the thickness of a pizza. But then it's kind of folded inside the skull. So it's just a few millimeters thick. And if you, just to give you some rough orders of magnitude, in every cubic millimeter of cortex in particular, but also of other structures, you would find approximately 100,000 neurons and about 1 billion synapses. And so, and this is in mammals. In vertebrates in particular, these numbers will be different. So if you divide, in fact, dividing these two numbers is instructive because it tells us that each neuron has approximately 10,000 synapses, which is a pretty huge connectivity. And in fact, there is a whole branch of neuroscience which is to try to understand first how many different types of cells there are in this circuit. And we still don't know actually the the exact answer. So there are many different cell types, as you can see on this plot. And also, how are these cells connected together? Right? So that's a major endeavor in neuroscience. So here, in this course, I will ignore this complexity, most of it. At some point, though, there's something really important, is that there are two main classes of cells in the brain. Some are excitatory, so they tend to activate their targets, and others are inhibitory, so they tend to inhibit their targets. And in this diagram, the red cells are excitatory and the blue cells are inhibitory. But as you can see, it's more complicated. The red cells, there are different cell types here. This is the pyramidal cells, because there are pyramidal cell bodies. These are the spiny stellate cells. And then for the blue cells, it's even worse. You have a huge diversity. You have basket cells here, bipolar, double bouquet cells, Martinez cells, and so on and so forth. And they all have different morphologies and they connect to other cells differently. As you can notice, they are pretty much, all of these cells connect to lots of other cell types, but typically not all, except pyramidal cells connect to everybody else. But internals tend to be more selective. They tend to connect to some other cell types than others. And we don't really understand, we still don't really understand the logic of this particular wiring. Okay, so let's just look at zoom in further. So this is a cortical pyramidal cell. Cortical pyramidal cells make up 80% of the cell in our cerebral cortex. So they are very important cell types, the most numerous cell type in the cerebral cortex. So as you probably know, there are basically three parts to a neuron. To simplify, there is an input side, which are the dendrites. So this is the apical dendrite that then ramifies. You have basal dendrites here coming from the sides. This is the input side, and so that's where neurons receive information from other neurons. And in pyramidal cells, these informations come at, so this is a zoom of one of these dendrites. You see that there are lots of little spines coming out of the dendrite, and that's where synapses are formed. I will zoom in further later. And if you count the number of these spines, you find also a number roughly of over 10,000, which is consistent with this division that I made a few slides ago, right? So again, these numbers are approximately 10,000 inputs. And then you have the soma, so the cell body, where you find the nucleus, of course. And then you have the axon, that's the output side. And that also branches, and the idea is that you connect many of the neurons nearby. And for now decades, yes.
So on the previous point, there's 10,000 synapses, but that doesn't necessarily mean that they're connecting to thousands of neurons, right? Like they can be connecting to just, 100 or fewer.
Yeah.
I don't know if there's actually any good evidence either way of how many they would be.
Yeah, thanks for raising this point. So you're right. So in fact, for a given pair of neurons, often there will be more than 2 synaptic contacts for a given pair. In fact, in cortex, for nearby neurons, these numbers can go up to 4 to 8.
Mm-hmm.
So in fact, so when I'm saying that there are 10,000 synapses, that might be, in fact, only about 2,000 presynaptic neurons.
Yeah.
But yeah, 2,000 would be more like a lower bound, because this number is 4 to 8 is like for really nearby neurons.
Okay.
But yes, very good point. So here, that's why I'm being quite vague and just say thousands without having a true factor. Right, so now, electrophysiologists basically can record the electrical activity of neurons by using electrodes. And if you record a neuron, so that's a cartoon, but basically if there are inputs, if the neurons is receiving inputs from other neurons, you see that the membrane potential of the neuron, which is typically hyperbolarized, this membrane potential will fluctuate basically as a function of the kind of the inputs that the neuron receives. If it receives an excitatory input, then the brain potential goes up. If it receives an inhibitory input, it will go down. And so it will kind of fluctuate up and down, and maybe there are synchronized excitatory inputs that arrive at some point in time, and so that brings the membrane potential up. And then what happens is that there is this dramatic event that happens, which is called an action potential. So if the membrane potential is above some threshold, which is typically about 15 millivolts from the resting membrane potential of the neuron, then there is this action potential that will then propagate down the axon, that will travel down the axon and then activate basically other target neurons. And in fact, basically, this is the action potentials is basically the only information that leaves the neuron. All these, the membrane potentials, so all these sub-threshold membrane potential is not something that the postsynaptic neurons have access to. They only have access to this so-called train. This is called the train of action potentials. And you can think about it as a kind of train of, it's often modeled as delta function, because they are very short, typically about 1 millisecond. All right, so let's summarize some basic electrophysiological properties of neurons. So this is a recording of a neuron in a brain slice. So where basically there are no inputs. The slice is completely silent. So you can maintain these neurons alive, and you see that they are the stable resting membrane potential, which is typically at -70 millivolts. And this is because of concentration gradients of various ion types. So there are more sodium ions outside the cell than inside the cell. And potassium ions is the reverse gradient, etc., etc. I don't have time to go into these kind of details, but basically that leads to a hyperpolarized resting membrane potential. But then what you can do is, using an electrode, you can inject a step current. And what you see is that if you have a small step current, then the neuron will just go to a new equilibrium, and it will go exponentially to that new equilibrium. But if you increase the amplitude of the step current, then you start, so there is a bifurcation, basically. You go to having a stable fixed point to now something that looks like it goes to a limit cycle. And one, in fact, one of the trends of research in computational neuroscience has been to understand the kind of bifurcations that lead to periodic firing. So there is a classification of different types of bifurcations that people have derived, which are essentially bifurcation from stable fixed points to stable limit cycles. Right, so that's basically the basic, all neurons more or less work like this. Last piece of information that I'd like to tell you is that the more current you inject onto a neuron, then the more action potentials will be produced per unit time. So the firing frequency will increase. And this is a summary here for one particular neuron. And these different curves basically shows the instantaneous frequency for different interspike intervals. So that shows that basically there is a phenomenon here called spike rate adaptation, so that the frequency tends to slow down with time if you have a sustained current. But that's something I won't really tell you much about later. What I want, just the only thing I want you to focus on is that basically there is this curve called the FI curve, so for frequency, current to frequency function that relates the input current of a neuron to its instantaneous frequency. So sometimes called transfer, and it's typically a monotonically increasing function of the input current. All right. So how do people model neurons? And so I just show in this slide basically three main classes of model neurons that people have studied, again from the simplest. And it's actually the three really simplest ones. And the list there would be approximately 100 bullet points down here, but they would go up to models that are described by thousands of coupled, highly nonlinearly coupled differential equations where you model spatially extended neurons using compartmental models and you describe in detail all the ion currents that are present in that model. Here I'm just cutting down the list of just 3, which are essentially the simplest models that exist. Okay, so the simplest one is the binary neuron. So the binary neuron, the idea is that at any given point in time, if you have a short enough bin, temporal bin, neuron is either emitting a spike or not, right? So it's pretty binary at a really short time scale. And so, and the idea is basically that you have the variable SI, which describes the state of neuron I, is either 0 or 1. And then these neurons typically have some kind of update rule where at time t plus dt, the neuron will be active depending on whether the synaptic input at time t was above some threshold or not. Theta here, capital theta, is a Heaviside function. So it's 1 if the argument is positive or 0 otherwise. However, now if you look at a longer time scale, in particular, if you come back to this, plot here, we see that in fact, if we think about the firing frequency of the neuron, this firing frequency seems to vary continuously as a function of the current intensity. And so, for this reason, people also came up with a so-called firing rate neuron. So in the firing rate neuron now, neurons are described by their firing rates, and now this is a continuous variable, which is of course has to be a non-negative variable because it's a rate of action potential firing, and it has typically some maximal firing rate because neurons have a refractory period, so they cannot fire at more than maybe a couple of hundred action potentials per second. And a popular formulation is this very simple equation where you have a first order differential equation, so the temporal derivative of the firing rate, so you have here the time constant of the rate dynamics, which is typically maybe about 10 milliseconds. And here you have some kind of decay term, meaning that in the absence of inputs, the rates will decay to 0 exponentially. And then it depends on the input through this nonlinear function phi. And this function phi, of course, is nothing else. It's basically this, correspond experimentally to this, to what the experimentalists measured as this Fi curve. And it's often taken as a sigmoidal function, right, for simplicity. So, now, slightly more realistic are models that describe the dynamics of the membrane potential, and then that describe the generation of action potentials. And essentially, the simplest model in that category is the so-called integrate and fire neuron. So now, in the integrate and fire neuron, we have the membrane potential VI that's governed by this simple first-order differential equation. And again, you have tau here, which is now basically the membrane, called the membrane time constant, and it's again 10 to 20 milliseconds. And you have here the so-called leak current, so in the absence of inputs, the voltage goes to 0 here. Well, here basically we set the origin of the voltage to the resting membrane potential, and then we have the synaptic inputs that drive changes in the membrane potential. And then in this model, you add by hand a threshold. So essentially, in this model, the membrane potential goes up or down depending on whether the neuron receives excitatory. So excitatory neurons with excitatory inputs will bring it up, in between inputs will bring it down. So it fluctuates depending on the timing of arrival of these action potentials.
Somebody can switch on the light to when Nicolas is writing, otherwise it's impossible to see through this. So it's, the light is there. So,
Can you see now?
yeah, maybe you can use maybe a blue pen, it would be. There is one.
I see only a black one.
Okay.
I don't know where are the other ones.
They are on the desk? In the boxes?
So this is the long-range control. This is better.
Better.
And then at some point it hits threshold, and that defines the time of an action potential. So here, action potential is sent to other neurons, and then you have to reset. In this model, you reset the voltage. This is another parameter of the model. You can reset it at some arbitrary voltage. Of course, it has to be below the threshold, and then it starts again, and then at its next action potential. And it's a very popular model. It's the simplest spiking neuron model. It's much simpler than models like the Hodgkin-Huxley model that describes action potential in a much more realistic fashion. And in spite of this simplicity, it has been shown to actually describe pretty well some a number of features of real neurons. For example, in the presence of noisy inputs, you can fit very well the transfer functions. You can measure the instantaneous frequency, the frequency of the neuron as a function of intensity in the presence of noise, and you can actually fit this function very well with the transfer functions that you can compute in weakly development neurons. Okay. Any questions at this point? Note that in this course I will be mainly focusing on binary neurons and rate neurons for the sake of time and simplicity, but in fact there is a lot of activity in the field that's devoted to networks of integrating fire neurons in particular. So what kind of dynamics networks of integrating fire neurons can produce? For lack of time, I don't have time to talk about it. All right, so now that we have seen how neurons basically work, let's see how they communicate with each other, and they communicate through synapses. And so now this is a picture where now instead of having just a single pyramidal cells, now we have two pyramidal cells that are sitting next to each other. So these are actually reconstructed cells from an experiment where they were first recorded in a slice and then they were reconstructed. And again, you see these amazing dendritic and axonal harmonizations. All these lines here are just these two neurons. No other neurons are shown here. And you see that it's so branched that basically these two cells, and that comes back to the question that you asked, they can actually contact each other at a number of different locations. So here these circles indicate the points of putative synaptic contact. So these are points where the axon of one neuron touches the dendrite of the other neuron. And so there might be a synapse. They can't be sure from those experiments that there is a synapse exactly here. Right? But there are these 5 putative synaptic contacts in one direction and 5 in the other direction. In fact, it's been estimated by these people, so this is Henry Markram and collaborators at EPFL, that this neuron can actually touch the dendrites, or essentially all other pyramidal cells in the neighborhood of size approximately 100 micron around it. So, and there are many cells. So let's make a zoom up. So what one of these done, right? So that's what I already mentioned, but it's a little bit more, we see a little bit better now. We see all these dendritic spines here of one neuron, one postsynaptic neuron, and we see axons of a number of presynaptic neurons. And then sometimes you have these synapses that are formed into the two. And basically, to describe in just 1 minute, very briefly, what happens at synapses, you have action potential traveling down the axon. At that point, cascade of events happens, so calcium increases, that leads to vesicles fusing onto the membrane here, releasing neurotransmitter, diffusing onto the cleft, binding onto receptors on the postsynaptic side. When they bind onto receptors, that opens the channel and ions can pass through and depolarize or hyperpolarize the postsynaptic neuron, depending on the type of ion that passes through. So positively charged ions like sodium or calcium, they polarize, and negatively charged ions like chloride will hyperpolarize. So that's a very quick description of what happens at synapses. And again, electrophysiologists can just record these single synaptic currents by having basically recording in a silent slice. Now you have two so-called patch pipettes. You see these two neurons, and then you can trigger action potentials in one of the neurons, and you can check what happens in the other neuron, right? And so in this case, from cell 1 to cell 2, well, nothing. So practically no synapse present here, and then you trigger spike in cell 2. Oh, you see something like this. This is called EPSP for excitatory postsynaptic potential. So here, clearly, there is a synapse connecting 2 to 1, but not in the other direction. Okay, so this is a unidirectional pair of neurons connecting unidirectionally. Sometimes you have synapses in both directions. And we'll come back to this issue of basically related to the degree of symmetry of the network in the last lecture. But we'll talk about, the issue of symmetry comes up quite. We'll come back quite often in this lecture. All right, so how can we model now synapses? So now let's focus on this. So I had this I of t, where the synaptic impulse to neuron I. Well, in networks of binary neurons, the synapse is just a single number, right? So the synapse connecting from neuron J to neuron I is Jij. Note that Jij is really a physicist notation, so that's kind of a history coming from a physicist working on spin systems. John Hopfield model had Jij as a notation. Most non-physicists in the field would call them Wij for weight, synaptic weight.
Are synaptic weights here just positive or negative, or are they strengths as well? Is Jij like ones and zeros?
Yeah, so we'll come back. So here I'm not specifying.
Okay.
But yeah, I will describe the differences between different models in terms of whether they are analog, discrete, sign constrained. So I'll talk a lot about this.
Okay.
At this point, it's just a number. Of course, if it's positive, it's excitatory, negative, neutral.
Yeah.
But in fact, the first models I will tell you about, there will not even be a constraint of the sign. Like in the Hopfield model, the synapses can go from positive to negative, which is of course, totally unrealistic picture. Okay, so in a rate model now, again, synapse is just a number, Jij, right? And so now we have basically describe the full. Now we have a full description of the network of either binary neurons or rate neurons. We see that the synaptic currents are the same in both cases. It's just the dynamics of neurons which is different. And here I also don't include external inputs that come from outside the network, but be included as an additional term. In integrated fire neurons, there is now one typically also includes describe the whole time course of these postsynaptic currents. And so the idea now is that, so here you see that in the synaptic current you have a single sum, which is a sum of the presynaptic neurons, J. Now you have a double sum, where this is a sum of the presynaptic neurons, Jij, and then you have an additional sum, which is about the the spikes of the presynaptic neurons. So it's sum of the K. So this function, so TJK here is the kth action potential of a neuron J. So if I come back to my little cartoon of the integrated fire neuron here, so this is basically T1, T2, and then this will lead to so this function G will be something like this, that with currents that are function G of TJK typically looks like this. And people have used a variety of different types of functions. The simplest one would be just a delta function, but we can use also basically any function we like, especially in simulations. So difference of exponentials can fit data relatively well, particularly.
One question.
Yes.
Just for in the rate neurons, it is only taken in account like how fast are you spiking, but not when exactly are you spiking. So in the input, what would is there any way to take in account, for example, two inputs that are synchronous or asynchronous? Because I think that would make a difference, right?
Yeah, you're right. So that's a limitation of the rate model that you kind of ignore. Essentially, the underlying logic of rate neurons is to look at the slower time scale than the time of generation of single action potential. So in a rate model, though, you can still have synchronization between two neurons. That's, for example, simultaneous increases in the instantaneous firing rate of both neurons. If you have a transient input to two neurons that drives their rate up, that will provide some synchronous inputs to the postsynaptic neurons. Right?
So rate neurons and to be understood like something that where you would be averaging for a long time in order to have this rate and not as instantaneous rate of fire.
Well, I mean, this is here the rate is a function of time, right? So it's not necessarily and this tau can be also something in the order of 10 to 20 milliseconds. The question, though, is whether this is a good approximation. I mean, the crucial question here is whether this is a good approximation of the dynamics of these kind of models. Now, this is a question that's been addressed by many people in the field. Like, how can you reduce the dynamics of a network of spiking neurons to a rate model? And typically, you can't really. I mean, in general, you can't. The dynamics of this model, you can't really reduce it to this because, well, I mean, this is just too complicated. But there are some limits in which you can do that. In particular, there is a limit when there is a slow variable somewhere. Let's say you have a slow synaptic time. Let's say this Jij decays on a slow time scale. Then on that time scale, you can reduce, you can do kind of separation of time scales, and on that slow time scale, you can reduce it to a rate model. There is another limit which is a little bit more complicated to explain, where even on a faster time scale, when you have the limit when you have lots of noise, so let's say your current is very noisy here, actually the rate model can provide a really good approximation of the dynamics. But not for this particular model. You have to move to another model which is called an exponential integrated fire neuron, and explaining why that happens will really take a really long time. But yeah, that's a very good question that for lots of theorists have tried to understand the relationships between these two classes of models. Okay, so since one crucial feature of synapses is that synaptic weights are not constant in time. In fact, they are changing on different time scales. Actually, they are changing on relatively short time scales of like hundreds of milliseconds and 1 second. That's called short-term synaptic plasticity, which I won't describe here. What I will be really interested in is here is the long-term, so-called long-term synaptic plasticity. And the idea why people are really interested in long-term synaptic plasticity is because it's thought to be the basic mechanism by which we learn and store information. And I'll explain this in more detail in a few slides. But now let's just describe this basic phenomenon. And so we'll come back to this slide with these two neurons that are synaptically connected. So there is a whole branch of the literature in electrophysiology where people try to induce plasticity in the synapses connecting these two neurons by activating the two neurons in different ways, right? And so typically that works in the following way. So first you check that you have a connection, so you have a pair of neurons. Most pairs, I mean the connection probability between these two neurons is only about 10%. Okay? So the probability that you actually see is about 10%. And that seems actually contradictory to a statement that I made earlier, that actually there are putative synaptic contacts essentially 100% of the time for me or myself. But I'll come back to this paradox in the last lecture. So now, for now, let me just mention that basically 10% of the pairs you see a connection. And so it looks like this. So you have this, so you trigger action potentials in one neuron and you see the EPSPs in the postsynaptic neuron. This, by the way, shows this short-term synaptic plasticity. You see that this amplitude here is much more than this one, and it decays. But now let's focus on the first EPSP. So now what you can do is, so first you measure the baseline EPSP. So this is done in the experiment, so here this is in time. So each time you trigger an action potential, you measure the baseline, and then you see that it's very noisy, because you see these fluctuations in amplitude, and that's a general feature in the nervous system. Everything is really highly noisy. It's something to keep in mind. And then at some point, you not only activate the presynaptic neuron, but you activate also the postsynaptic neuron. And in this experiment, they activated systematically 10 milliseconds after the presynaptic neuron. So you pre-post, so you have the presynaptic neuron and the postsynaptic neuron, and so the protocol is like this. And you do it something like 60 times. 60 times. And then what happens, after you do this, something like 60 times, now you see that the synapse is now stronger. So this is this basic phenomenon of synaptic plasticity, that you've changed the weight of the connection between these two synapses. And this shows the comparison between the average postsynaptic potential before and the average postsynaptic potential after. You've almost doubled the weight of the synapse.
Sorry.
Yes.
So what you're discussing also in general, is it valid like across different like biological entities? It's something which is mainly true if you look at C. elegans, Drosophila, and a monkey, or are you looking at a specific?
Yeah. Very good question. All the experiments I'll be showing you are in rodents. In fact, rats or mice in the cortex of rodents. We know very little about plasticity in animals like C. elegans. We start to have more information about insects. In fact, this phenomenon is called spike time independent plasticity. So this has been demonstrated in insects actually. So this is apparently holds true in insects. And yes, so there are, it's really very, there are a few animals where this has been done. It's still too early to say how general this is, I would say. In rodents, this is very robust in many cortical areas, in the hippocampus. And there are other structures where you have different synaptic plasticity rules, which I will mention briefly. So this is not universal by far. But people believe that it happens in different species. Yes, that's true. Okay. So from like tons of experiments, a real brief summary of what can modify synapses, and there are let's say, two different classes. So we're coming back to this issue of firing rate versus spike timing. And so the experiment, let's start with the right panel. This is the so-called spike timing dependent plasticity protocol. So you're activating a pre and post-synaptic single action potentials, pre and post, with a fixed delta t, the time difference between the two. And when delta t is negative, so post before pre, then typically synapse goes down. When pre before post, synapse goes up. So that's called the spike timing dependent plasticity protocol. And notice that this is essentially the so-called, sometimes also called the causal pairing, because the pre is firing before the post, so maybe the pre took part in firing the post, and if it does, then the idea is that it tends to increase the synapse. And we'll see the consequences of the network level of this type of plasticity later. But in fact, you don't need these systematic relationships between the timing of the spikes to induce plasticity. There are in fact lots of other experiments where people just, okay, let's just increase the firing rate of these cells. And this is an experiment where now they actually use random delta t's for each pair of spike. They draw delta t randomly from some uniform distribution. And what you see is that, well, you tend now, what happens is that at when both neurons are firing a lot, high firing rates, then you have potentiation. When they're firing at kind of lower rates, then you see depression. So, in other words, the change in synaptic strength depends on the spike trains of pre and post-synaptic neurons in a kind of complicated way that depends both on firing rate and spike time. And there is a lot of people have tried to describe this data quantitatively using various types of models, which I don't have time to explain. But I will just here, I just want to emphasize that this has given rise to two different trends in the theoretical literature. On one side, you have the firing rate-based models where the change of synaptic strength depends just only, it depends on the firing rates of post-synaptic neurons and pre-synaptic neurons only, and these are just firing rates. On the right, you have now rules where the basic variables driving plasticity are the spike times of neurons, Tij. The Ti's are the, and Tj's are the spike times. And a simple rule is basically a rule in which they depend on the spike times only through the difference of the, through the differences between Tjk minus Tijs. But there are also more complex models that take into account kind of higher order terms and so on.
And is there some result that if there is only pre or only post, then you don't have plasticity? Because it seems that you insist on the fact that the protocols are always using both.
Yeah, so what you see typically is that in fact you need both. You need a coincidence. Yes, that's a very good point. And we kind of understand why, so I'm not planning to tell you anything about the biophysical mechanisms of, but interestingly there is a receptor, a particular type of receptor called the NMDA receptor, which is basically has been shown to be a biophysical implementation of a coincidence detector, because it needs both pre-synaptic activity and post-synaptic activity to open. And it's in fact it's crucial. If you block this receptor, typically you erase all plasticity with these experiments. So okay. So this gives rise to plasticity rules. So sometimes, these go under these kind of equations, go under different names. People talk about plasticity rules, learning rules, where the idea is already that it helps you to learn something. But they are part, in fact, of just one class of learning rules called unsupervised learning. So in unsupervised learning, the idea is that synapses have only access to the firing rates of the two neurons that they connect. That's the only information that synapses have, and they have to basically evolve just based on that information. So that's called unsupervised learning. There are other types of learning that have been studied extensively, which unfortunately I won't have time to tell you about. I'll tell you in the last lecture, I'll tell you a little bit about supervised learning. reinforcement learning is learning where the plasticity is gained by reward. So the idea is that animals just explore the world, and at some point they get some rewards, maybe some food. And the idea is that you want to reinforce actions that you learn lead systematically to rewards. And this is thought to come, reward is basically linked to a neuromodulator called dopamine that's released by some structures in the brain like substantia nigra, and the dopamine basically is released in target structures, and this reinforcement learning would happen in those structures. Note that basal ganglia is involved in selection of actions, so that makes sense. You want to select actions that will maximize the likelihood of future reward. And there is a whole field of research on that particular aspect of learning. Supervised learning is yet another form of learning where now you receive some error information. Suppose that you're trying to learn a particular skill, like sailing, for instance, right? So you're doing something, and at some point your boat flips and you fall into the water. So you clearly did something wrong. And the idea is that this error is actually encoded in some brain structure, in particular inferior olive, and communicated through the cerebellum. Remember that cerebellum is the structure that's associated with motor learning, and that you are using this error information to modify basically your input-output transfer function. So next time, maybe you'll be less likely to make that error. Okay, just give it some time. All right. So briefly, so at this point, I told you about the components of networks, so neurons, synapses. What about how do people model networks? And of course, so brain networks are incredibly complicated, and we have only partial information about most of them, again with a few exceptions like C. elegans that I mentioned at the beginning, but that might not, the conclusions from C. elegans might not really carry over to more complex animals, because neurons in C. elegans are connected to just a few of other neurons in C. elegans brain, and certainly not 10,000. And so that's how a cortical network would look like. So it's really a very dense forest of cables, axons, and so most of the volume actually in the nervous tissue is just cable, axons and dendrites that are used to connect it. So again, we can go from the simplest to the more detailed. We can start with a fully connected network. That's where I will start tomorrow, like the Hopkins model is fully connected. But of course, that doesn't take into account that I already mentioned that if you record pairs of neurons, you see that there is only, you find a response, a synaptic response only in maybe about 10% of cases. That's a typical connection probability for nearby neurons. So then the next simplest model that takes this into account would be like an Erdos-Renyi network, like a completely random network with some prescribed connection probability. And of course, but that doesn't take into account the fact that you have multiple neuron types, for instance, excitatory inhibitory, or you can have multiple subtypes. You can have neurons, we'll come to this later, neurons can be different in how they respond to external inputs, so that leads to a very much more heterogeneous networks, and basically these different types of neurons might have different connection probabilities, and in fact they do, that's been shown experimentally. So now you have a model in which your connection matrix is no longer specified by a single connection probability, but you have something that's sometimes called stochastic block model, where you have a network where, depending on the identity of pre- and postsynaptic neurons, you will have a different connection probability. So your model is specified by a matrix of n by n connection probabilities, where n is the number of populations. And last, of course, so far I've ignored space, and of course these networks are embedded in space. Of course, the neurons that are close together are more likely to be connected than very distant neurons. In fact, connection probability decays typically on spatial scales of a few hundred microns. However, that doesn't mean that very distant neurons cannot be connected. In fact, there are also very long-range connections in brains. And so that leads to the picture of at a really large scale of something that might be closer to a small world network because of this mixture of local and long-range connections. However, here I'm really focusing on models that are, the idea is to focus on networks at the local scale, so at the scale of maybe 1 millimeter or so. So I will completely ignore the spatial structure in these lectures. All right. So now, just a few very brief snapshots of first how, before talking about how information is stored in brains, let me just mention briefly how information propagates in the brain. And this is kind of I think it is a useful picture to first visualize what happens when, from the moment you perceive a visual stimulus and then you generate an action in response to that stimulus. And it's kind of, I think it's really instructive to see all the areas that are involved and also how fast information propagates, because as you can see from the time you see something and the reaction of your muscles, they're only about 200 milliseconds. Well, actually these numbers are from macaque monkeys, so we are a little bit slower. By maybe, and this is actually a macaque monkey, you might not have noticed. Right, so basically that tells you that information will propagate very fast in your visual system as you perceive a stimulus. And these times are actually derived from electrophysiological experiments where people measure the activity of these neurons. They present a particular visual stimulus and they look at the latency of activation of neurons in those structures. And you see that essentially they are basically the times increase, latencies increase by essentially about 10 milliseconds from one structure to the next. Visual information is analyzed in these successive layers, where here we have very simple types of analysis, like V1 neurons are sensitive to simple features like edges or corners. And then this is elaborated, there is this hierarchy where progressively neurons are selected to more and more complex features. Eventually, so there's a big chunk of our visual system called the inferior temporal cortex. I will come back later to this structure, where you have neurons that are selected for faces, for instance. You have neurons that can be selected for chairs, tables, various objects, screwdrivers. So the idea is that basically when these neurons become active, they kind of signal to the rest of the brain, hey, there is the face of Claude's face. Claude is here now because I recognize his face. And I'll come back to this later. And this happens very fast. You see that it's about 100 milliseconds. Then it goes to these other brain areas, prefrontal cortex. Now, this is the area most classically associated with decision making. And so, for instance, again, this idea that you have a visual stimulus coming at you, maybe a ball coming at you, you have to react, maybe do something to catch it. So the decision is taken in the frontal cortex. And then the decision is sent to the motor cortex that sends the motor command to spinal cord to the finger muscle. And yeah, so one, of course, and particularly in the field of computational neuroscience, there is a whole of activity trying to understand the nature of how information is encoded in all these different areas. There's the nature of the neural code. How can information propagate so quickly, and so on and so forth. But that's really not what this set of lectures will be about. Rather, here it will be how is information stored. So information can propagate in these different brain areas. Now, how is this information stored? So this is now. So this is really the last part of this introduction. And this is a cartoon that explains kind of try to give an intuitive explanation of what happens. So So suppose that this is a network in IT cortex. So the IT cortex receives this visual information which has already been pre-processed by all the earlier areas. And let's say you have a new stimulus, let's say you've never seen this animal before, and this will trigger some activity in this particular structure. Or this could also be the hippocampus, which is even later in this visual hierarchy. So the idea, so the first step, of course, is you need to have activity in that structure that's driven by the visual stimulus. Now the second step is that this activity now in turn leads to changes in connectivity. And that's the storage step, that you're kind of imprinting in a way the stimulus in your synaptic connectivity. Here, what I've represented here is sometimes what is sometimes called the Hebbian synaptic plasticity. So Hebbian synaptic plasticity from the Donald Hebb, a Canadian psychologist that wrote this very influential book in the 1940s, and there was no experimental data on synaptic plasticity at all at that time, but you had to wait until the 70s for this data to come, and he hypothesized that if two neurons are active together, they must reinforce the strength of the synapses between them. And they also propose also the causal version of this principle, that if the neuron is active before another neuron, then also maybe there should be an increase in synaptic strength. So here is the version where you don't have the temporal aspect, and it's sometimes referred to as neurons that fire together, wire together. This is the basic Hegel principle. And we see that here, this group of neurons have been active together, and you have strengthening of synapses that connect neurons. And the idea is that now the stimulus has disappeared, but there is now a trace in that network, right? That maintains a memory of this stimulus. And of course, you could have other stimuli activating different populations of neurons, and leaving again traces in this network. So in this picture, connectivity matrix will be seen as a superposition of traces left by external stimuli that are modified. So that's the basic idea, and this has been implemented in countless models, some of which I will review in the next lectures. And I will review in particular two big classes of models. So in one class of models, the idea is that, so suppose that you have a group of neurons that are active together for some amount of time, and with this Hebbian mechanism, so the idea is that you strengthen connections, but you will strengthen connections in both directions in this group of neurons. So we will tend to generate, the connectivity matrix will tend to be pretty symmetric, right? And that's consistent also with this data that I showed earlier, where if in fact, I showed you that if two neurons fire at high rates together, then you tend to reinforce their synaptic strength. And the idea of this whole class of models that I will start describing tomorrow is that this leads to new fixed point attractors of the dynamics of the network. So now there will be a configuration of the network where these neurons are active and the others are silent. This now becomes a stable fixed point of the dynamics. And so now this is a stable fixed point, and the idea is that then, so one benefit is that the network can now maintain the identity of this stimulus in short-term memory. So we present the stimulus, these neurons get activated, and then the stimulus is removed, but these neurons keep, they stay active. And that allows the network to basically keep this stimulus in short-term memory. And that's actually consistent with the data that I will review later, where so the prediction of this type of model is that some neurons should keep something that's called persistent activity. So they are activated by a stimulus, here shown transiently, and then they stay active even after the stimulus has disappeared. So that's one class of models. This will be in particular in the next two lectures. I will go through these models of this type. Another class of models now give rise to radically different types of dynamics. And now the idea is that we have a more dynamic picture where suppose that you have a more temporally varying stimulus. This temporally varying stimulus activates different groups of cells in succession. And now the idea is that what we use now is the fact that if you have this spike timing dependent plasticity where this mechanism will strengthen connections only in one direction, the pre before post direction. So in this case, the connection in this direction will strengthen, but the connection in the other direction will actually weaken. So we'll develop a strongly asymmetric connectivity. And the idea is that now, potentially, what can happen is that when after you structure the connectivity matrix in this way, you activate these neurons, you will be able to retrieve the whole sequence from just activating the first group. So the idea is that this network will store sequences, and that's this phenomenon is seen in other experiments that I will describe in the fourth case. Okay. I think I still have time. When should I? I'm actually almost done, but just to 15 minutes. I'm still okay? Yeah. All right. I just want to show you some basic experiments in evidence for this phenomenon of attractor dynamics. So the fact that networks have these stable states. And they can use them to store information in shorter memory. So this is so many of these experiments were done initially in macaque monkeys, and the idea again is because of the similarity of monkey brains with our brains. So this is the typical experimental setup with the macaque monkey sitting in front of the computer screen, and in these experiments, you can show different types of pictures to the monkey. Monkeys learn very easily, lots of different tasks, and at the same time, you can record the electrical activity in specific brain structures. And this is an example of such a recording. So this is a recording in this area called inferior temporal cortex. This is when one particular image is shown at time t equals 0. And this is one neuron. No, these are different presentations of the same image. Here you have 10 different presentations of exactly the same image. The sensory stimulus is exactly the same. In each of these rows, so this is the activity of this neuron, and each of these little ticks is an action potential of this particular. So, what do you see? So first, what one sees is that even in the absence of visual inputs, neurons in cortex are active. This is called spontaneous activity or background activity. This is a ubiquitous phenomenon in many brain structures. And still not clear why this has a functional role. Now this spontaneous activity, as you can see, looks totally random, right? Different trials, this is like extremely different. There is this randomness, apparent randomness both across time. In fact, this is similar to, but not exactly, it has some similarity with the Poisson process. If you compute Poisson distribution of these interspike intervals, these typically are exponential tails as in a Poisson process. This is also extremely valuable from trial to trial. Here you have a trial with a pretty high fine rate. In fact, typically the variability from trial to trial is even higher than the Poisson process, consistent in fact with the doubly stochastic process. So the idea is that at each point in time, a simple model for neuronal activity in background in spontaneous activity is that at each point in time, you have a rate that itself a random variable, a slowly varying random variable, and then the neuron is basically emitting spikes as a Poisson process with that specified rate. As you can see, you see that here it's a pretty high rate, here there are no spikes. And there is again a whole branch of the field which is about trying to understand like how you can get this kind of dynamics in networks of spiking neurons, which I won't talk about. Okay, so that was this part. Now the second part I want to mention is, which I will be focusing on, is the fact that this particular neuron, the firing rate increases dramatically approximately 80 milliseconds after you see the stimulus. So this is this latency, so the 80 milliseconds is the time from the signal to come from the retina to this particular area. And in this case, it kind of increases a lot and then decays this way. So this is a neuron that really likes this particular stimulus that has been shown. But typically, this is like a really strong response. This will happen typically less than 1% of stimuli.
Sorry?
Yeah.
One brief question. So the deep before, is it random or is it biological?
Yeah. Good. Yeah, very good question. This has been reported in experiments when the animal can actually expect the time of the stimulus to occur. I'm not sure in that particular experiment, to be frank, whether this has been shown to be significant. I mean, here it could well be something. I'm not sure this is a significant decrease from that. But yes, sometimes you have some significant trends when the animal can expect a stimulus to occur at a particular time. And there is a growing literature on these phenomena that are likely related to the fact that the animal kind of estimates time. So it has a basically estimate of time in its brain, and that actually modulates lots of processes in the brain. Okay, so this is kind of one of the simplest experiments to probe this phenomenon of short-term memory, or actually a form of short-term memory that's sometimes called working memory. So working memory is a form of short-term memory where you have to maintain something in active memory because it will be useful for some later action. Okay? Like let's say you're doing a calculation, you're using a lot working memory, right? Because you don't have everything in your there are lots of things that you have to maintain in your working memory in order to perform the different steps. So this is a really simple example where the monkey first, so these are the different stages of the experiment. There is an initial fixation. The monkey has to maintain the eye. To start the trial, you have to fixate here, so that he has to look at the central spot. Then an image is presented called the sample. Then the image disappears. Then you have a delay period, so there is no longer any information on the screen. And then this is a version of the task where two images are shown side by side. And basically, as you can see, one of the two is the same as the sample. So the animal in this case has to go and look at the choice, or maybe in some versions just to touch the so-called match, the picture that matches the sample. And so for this reason, it's called delay match to sample task. The idea is that it's a very simple experiment where you can probe the mechanisms by how the information about the sample picture is maintained across this delay period. And the animal gets a reward if it gives the right answer. Experiments have shown that you have neurons that might be involved in this maintaining the information about the stimulus in many cortical areas, actually, including IT cortex, vertical cortex, also some areas in parietal cortex. Okay. So it's not by no means located into a specific cortical areas. It seems to be a widespread cortical phenomenon. And just to show a few examples, so this is actually the first experiment by Joachim Fuster from UCLA at the beginning of the 80s, where he just used basic colors, so color screen. So here is a green color, here is a red color. This is a neuron that, when you present a red color as a sample, so this neuron increases its firing rate, so it's sensitive to color, this particular neuron. And then it maintains a high firing rate for something like almost 20 seconds. So for a neuron, 20 seconds is like forever. The basic time scales of synaptic, the time constants of neurons are like in the tens of milliseconds. Okay? So this is like a really long time scale that maintains this decline. But this neuron doesn't care about green. There are other neurons, of course, that will care about green. So you can think about it as each neuron picking some color because of probably the particular combination of inputs that it receives. Other experiment where this was done with abstract patterns, but that's really similar phenomenon. This neuron likes this particular pattern, but not others. And it also maintains this frame rate for a really long time. Yet another experiment with now pictures of objects. Very similar pattern. And note that in these experiments you can see that basically this is consistent with what you would expect for a fixed point attractor. Basically the neuron goes to some configuration, to some particular frame rate, and then it stays there for a long time. But of course there might be other explanations. So how can we test this idea? So the idea is that you'd like to make perturbations, right? So if you make perturbations, the idea is that if it's a stable fixed point, you have a small perturbation, you should go back. But if the perturbation is large enough, then you go away and you go to some different, maybe attractor. And this, we can't do that currently in monkeys. That's not doable. But you can do that in mice. So this is a recent experiment where, just to show you, just to conclude, a very recent experiment in mice where they first identified an area in mouse, in mice where they found this phenomenon of persistent activity. And this is really an amazing experiment where to identify the area that's responsible for the maintenance of information in this task, in short-term memory, they actually inactivated sequentially everywhere in the cortex. This is a view from the top of the brain of the mouse. This is cortex. This is cerebellum. This is the olfactory box. This is the front. This is the back. And they went on and they kind of inactivated using a technique called optogenetics. So with optogenetics, you can activate neurons using light nowadays, which is a really amazing technique. And if you activate inhibitory neurons with light, you're essentially shutting down the area because inhibitory neurons will just shut down everything. And you can go and activate, and basically they found that this particular area here, called the ALM, this was the only place in cortex where when they inactivated it, the mouse was no longer able to perform the task. So what was the task? So very simple task. These are mice, so you can't do the same thing as with monkeys, but here the idea is that there is a tone, high or low. Then there is again this delay. And then basically the mouse has to associate the tone with the response, which is either left or right. Okay? And the idea is it will lick left, the reward will be on the left if it's if it was a high tone, or a low tone will be on the right. And if it licks at the wrong location, there is no reward at all. And you see here example neuron. So this is a neuron that increases its firing rate during the delay, has a high rate for right, but low rate for left. And this is actually, this is the firing frequency is 5 per second. So this is the average over many trials. And this is the membrane potential of the same neuron, so they can switch. You can record using intracellular recordings. You can record the membrane potential. Now, the next part was to actually do this perturbation experiment. So that's the last thing I'd like to show you. And so for this, okay, I need to explain a little bit here what they're doing. Here they computed so-called a coding dimension for population activity. So you have, basically they are measuring the number of neurons, I forgot how many neurons, maybe over 100 or so. And so at each point in time you have the activity in this 100-dimensional space. And what they can do is they can try to find the vector in that high-dimensional space that maximally separates the trials where the animal has to lick right or left. And that's what they call coding dimension. Now at any point in time they can project this high-dimensional activity vector on this coding dimension. That's what they're doing here. So this is the activity projected to the coding dimension for the right trials. So this blue curve, and this is with the left trials of the red curve. These are the correct trials, with no perturbation. And we see that we have this nice separation that actually is growing in time. And that's again, there was this question, I think I made this point that there is this time dimension in many of these experiments. So that shows here that's related to the fact that the mouse is expecting to produce a response at a specific time. This disappears if the delay times are completely random and unpredictable. Then you no longer have this trend of perturbation. Now if you make a perturbation, so again they can perturb in different ways, but the optogenetic perturbation is really ideal, so they can basically, with the light, perturb the network, and they can vary the intensity of the perturbation. And sometimes you have, they perturb, but after the perturbation, the network comes back to the unperturbed trajectory. So similar with what you would expect with a system with a stable fixed point attractor. So basically the perturbation didn't leave the basin of attraction of that fixed point, so it comes back. Now sometimes actually, and in fact here they took only the correct trials. Now if they take the incorrect trials, basically you have this flip where now for the basically you can see that there is this switch between the red and the blue cases. So basically essentially the neurons that would code for left would not get activated while the correct response is right. Alright, so that's a very nice experiment. That's actually just from, this is from just 2 years ago. Alright, so that's just a basic, serves as a basic motivation for the topics that I will address in the next lectures. I just included in this slide, in these slides, a lot of references at the end, so I put it on the Dropbox folder. So this is basically, I just tell you very briefly, so I have a few for those of you who would like to learn more about neuroscience. Just give my selection of neuroscience textbooks. This one is really a kind of Bible of that have been used by many generations of neuroscientists. In fact, it was used to, people used to call it the Kandel and Schwartz. Originally the authors was Kandel and Schwartz, but now in the 6th edition the authors have changed. And this book is maybe at a little bit simpler level and less detailed, very condensing party. So this one goes more into the really single cell neurophysiology and single cell models. They are much more, these books are basically barely any equations. This book has lots of detailed biophysics of single neurons with also description of models and stuff like that. Okay, computational neuroscience, I have this huge list. I think I don't have time to go through this list, but if you wonder which book you would be best suited for you, I mean, if you're interested, I can give advice depending on what are your specific interests in the field. I think different books are better suited for depending on your interests. So there are books that are really more like for physicists, like Engel and Brunel and Statistical Mechanics of Learning. It's an old book by Danny Amit, but that's still quite useful, and others are really not specifically targeted to physicists and more generally to computational neuroscience. And then I have some bibliography about what I will talk about in the next lectures. So.